Okay, hello everyone, welcome back. Although we have a lot of activities to go through today, it's going to be a pretty brief video since some instructors did record themselves doing these activities. So I'm not going to really explain too much in detail the activities. I'm just going to give you guys a little bit of background so you understand the activities they demonstrate. Go ahead and share my PowerPoint here. And I'm going to start off here. Okay, so there's some concepts that you need to understand for the next activities. You're going to need to understand the diffusion. So diffusion is the movement of molecules from a region of high concentration to low concentration. And there are a few factors that can affect the rate of this diffusion. The rate of diffusion is directly proportional to temperature, and the rate of diffusion is inversely proportional to the mass of the molecule. And you will actually be seeing this in the experiments we conduct. You'll see that heavier molecules move more slowly than lighter molecules when this occurs at the same temperature. And your book also kind of explains diffusion in the real world and gives an example about how if you were to spray some perfume fragrances in a enclosed environment, the molecules don't really disperse, so that scent is more concentrated. However, when you spray like a perfume outdoors, the fragrance is going to dissipate rapidly, and you don't have that scent lingering around as much. So diffusion. So in order for the cell to communicate with the external environment, materials must be able to pass through the plasma membrane or cell membrane. So here I wrote plasma membrane, but it really depends. Plasma membranes are usually around organelles. Cells membranes are usually around cells. But essentially both of them are semi-permeable and both have to communicate with their external environment. I just sometimes use them interchangeably, and I don't want to confuse you because that's actually not correct. I shouldn't be doing that, but most people tend to do that. So the movement across the cell membrane or plasma membrane could occur through several mechanisms. So in this image here, I show passive transport. So passive transport or simple diffusion, it works great for smaller nonpolar molecules. No energy is required, and a substance diffuses down its concentration gradient from where the substance is more concentrated to where it is less concentrated. So if we were to look at this, this would be your extracellular environment or at the outside of the cell, and this would be your intracellular environment or inside of the cell. And as you can see on the left, you have these red molecules that are highly concentrated. So these molecules want to go to the right. They want to eventually reach equilibrium. So you want to have the same amount of red molecules on the left as in the right. So they're going to do this through, this shows like a pour, but it'll do this essentially through simple diffusion. It's just going to be able to pass through that cell or plasma membrane. So eventually these molecules, see here it's ricocheting off of the membrane. It didn't quite make it through. This one's able to make it through. This one's making it over here. This one's going that way. And they're essentially going to go back and forth until they reach equilibrium. So here the red molecules will be in the same concentration both in the extracellular environment as in the intracellular environment. So here what you're trying to say is that the red molecules are going to diffuse down their concentration gradient to reach equilibrium. So they're going to go from where they're highly concentrated to where it is less concentrated. So this top example is showing passive transport of one molecule across the plasma membrane. Down here in B, we show passive transport of two types of molecules. So as you can see, in the extracellular environment, you have a high concentration of this blue molecule. So the blue molecule wants to go into the cell, whereas this red molecule is highly concentrated in the cell, so it's going to want to go outside of the cell. Like I said, they're basically going to be going back and forth between the intracellular and extracellular environment until they eventually reach equilibrium. So both the blue and red molecules are present in about equal numbers, both in the extracellular environment as in the intracellular environment. So another way cells can move, okay, so sorry. So here on our left-hand side, this is what I just explained. This is the passive transport. It's just able to basically diffuse through this lipid bilayer or your plasma membrane or cell membrane. Another form of, another way molecules could go from the extracellular environment, so out here to inside the cell, would be through active transport. So active transport is represented over here. This usually will require some kind of transporter or receptor that's embedded in the plasma membrane. This is going to require energy. So energy here is represented as ATP. And as you can see, this molecule is found in, there's less of this molecule outside of the cell compared to inside of the cell. So this molecule should be wanting to go this way. It should be going from the inside of the cell to the outside of the cell. That's along its concentration gradient. However, in active transport, it's essentially doing the opposite. It's going against its concentration gradient. So in order for this, which is there's less of it outside of the cell, to be able to go inside the cell where there's a lot of it, it's going to require energy because the cell doesn't really like this. The cell membrane doesn't like this, and it's going to need help to travel across the cell membrane. If you guys want to look further into this, a great example would be the sodium potassium pump. This is found in the central nervous system. So for example, your brain. So you guys can go ahead and look that up on your own if you want to learn a little bit more. And then facilitated diffusion. This is once again going along its concentration gradient. So as you can see this orange triangle, there's more of it, this molecule outside of the cell than inside. So it wants to go into the cell. So it's going along its concentration gradient. However, it typically will still use a transporter receptor, and that is because this molecule will usually either be bulky or large, or it'll be polar, so it's going to be difficult to pass that the lipid bilayer. So it's going to go down its concentration, but this is for larger or polar molecules. So for example, if you had a molecule that wanted to go across a cell membrane, which molecule do you think would travel faster? Do you think water would travel faster or glucose? So remember, water is basically 2 hydrogens and an oxygen, whereas glucose is going to be a little bit more complex than that. It's going to be, I don't know, like 6 carbons, 12 hydrogens, and 6 oxygens that are going to 6 carbons, 12 hydrogens, 6 oxygens, yeah, that are going to need to get across. So obviously water is going to be a lot smaller, so it's going to be easier for this to travel across the cell membrane than glucose. Another example would be, would it be easier for glucose to get across a cell membrane or starch? So remember, glucose is a monomer or a monosaccharide of a carbohydrate, and starch is a polymer. It's essentially a bunch of alpha glucose molecules combined together. So once again, obviously it'll be easier for glucose to pass the cell membrane because it's a lot smaller than starch. So another concept you'll have to know for your experiments is osmosis. So this is essentially the same thing. However, before we were just looking at molecules in general and any solute. Now we're going to be looking at solely water. So we're going to look at the movement of water molecules from high concentrations to low concentrations. So down here we have an image where you can see this yellow barrier is the depiction of a cell membrane or a semi-permeable membrane. And you can see your solute here is represented as a sugar molecule, these green dots. So your water's basically going to want to go from high concentration to low concentration. So you have low concentration here, high concentration here. So your water is going to actually go this way. It's going to leave the area of low concentration and go to the area of high concentration. So essentially what it's trying to do is dilute the area of high concentration so that it somewhat matches this area of low concentration. So right now, this area is like super diluted. This area is more potent. When the water is able to go to the area of higher concentration, it's going to further dilute this area. So eventually it'll be isotonic. So there's some important terms I have to go over. So solute in this example is going to be your sugar molecule. Your solvent would be your water. So water is actually a very popular solvent for most material. And your solute, it doesn't have to be a crystal or powder, but typically it's depicted as a crystalline structure or powder structure. But you could also have a like a viscous type of syrup consistency be a solute. That's also common. So something like that would be tween 20. And then another few terms we have to go over are hypertonic, hypotonic, and isotonic. So this area where you have a low concentration of solute, this would be referred to as a hypotonic solution. The area that has a high concentration of solute is referred to as a hypertonic solution. And when these areas are the same concentration, when they basically reach this equilibrium, it's going to be referred to as isotonic. So different cell types respond to these solutions differently, and it really depends on what kind of cell type you're looking at. So on the top, we have red blood cells or erythrocytes in animal cells or mammalians. And as you can see here, an animal cell is, or a red blood cell is happy under an isotonic solution. So water is able to come in, water is able to leave, and this is the condition an animal cell is most happy. In a hypotonic solution, you're going to have a huge influx of water coming into the cell. And this is going to cause the cell to lyse, or in other words, burst. So when you have a hypotonic solution, the water is going to rush into the red blood cell and it will burst it. And then in a hypertonic solution, when you have a higher concentration of solutes in the exterior environment, it's going to make the water from inside the cell want to leave. So the water from inside this red blood cell is going to want to leave the red blood cell and basically dilute the surrounding environment. And this is going to cause the red blood cell to shrivel up. So now in a plant cell, because they have certain structures, such as the central vacuole, the cell membrane, and the cell wall, they actually prefer a hypotonic solution. So this is why your leaves will be able to stand up like perky when they're in hypotonic solutions. So in an isotonic solution, the water and coming in is equivalent to essentially the water going out, and you will see that your plant cell will begin to wilt. It will look flaccid. It's not getting enough water. When you're in a hypotonic solution, there's going to be a lot of water rushing into the cell. And a plant cell has a few structures that allow it to retain this water. So the central vacuole is this structure that can just retain an absurd amount of water. And the reason that this cell won't burst like a red blood cell above is because the cell membrane will push up against this cell wall, and this cell wall is very thick. It's made out of cellulose and it's very rigid, so it's able to hold those components in. Remember, a animal cell that only has that cell membrane. It doesn't have a cell wall. So here it'll burst. Here it can hold that structure. So that's actually what your plants prefer. And then in a hypertonic solution, the water is going to leave the inside of the cell and try to dilute that exterior environment. And what's going to happen is it's going to start to shrivel up, and it'll actually start to undergo plasmolysis. So the cell membrane will pull away from the cell wall. So you'll have the cell wall still out here because it's so rigid, but then you see that the cell membrane starts to like pull in and shrivel up. So this is just kind of going over it again. You guys can kind of practice with these diagrams. It's just showing how the solvent is water, your solute is sodium chloride up here. You mix it up, you eventually dissolve these salt particles or molecules, and then you have, I guess, saline. You guys can practice with these images here to understand hypotonic solutions, hypertonic, and isotonic solutions. But very briefly, these brown dashes are your semi-permeable plasma membranes or cell membranes. And what you have inside the bag are these blue are your water molecules, and these purple are your solutes. So you have a hypotonic solution inside the bag. Compared to the beaker. So what do you think is going to happen? Obviously, you're going to have some water that's going to leave the inside of the bag, go to the beaker to be able to try to dilute this area. And then this is just the reverse. And then here you can also review this with water, like which direction water will go to. So if we were in lab, you would conduct activity 3. Here, essentially what you would do is very similar to activity 1. You would prepare a wet mound of the Elodea plant. So you would use forceps to take one leaf off of this freshwater plant. You would place it on your microscope slide. And then in this experiment, you would either put water on top of the pond water on top of the leaf and then put your cover slip on top of that, or you would expose it to a hypertonic solution. So you would put, I believe it's 10% sodium chloride solution on the plant and then place your cover slip on that. So if you went to the microscope to look at this, the leaf that you just put the plant water on, which is considered a hypotonic solution, you would see something like this. So if you remember, plant cells are much larger than animal cells, and they have this cell wall. These green ovals are your chloroplasts, and you would see something like this. They're very nice and plump. So essentially this is occurring. The piece of leaf that you put the salt water on top of, you would see plasmolysis occur. So essentially the cell wall would retain its structure as shown here. However, this cell membrane will begin to pull away and everything will shrivel up. And essentially what is happening is it's exposed to a hypertonic solution. It begins to undergo plasmolysis. So water is leaving the plant cell and trying to dilute that exterior environment. So what happens to the central vacuole during these conditions? Well, in this condition, the central vacuole is retaining all the water it can. It's looking nice and plump. In this condition, the central vacuole is essentially letting all sorts of water leave the plant cell. And why don't plant cells lyse when placed in hypotonic solutions? So why aren't they bursting when they get placed in hypotonic solutions like we saw in the red blood cell? And that's really due to two structures. It's due to the vacuole, which can retain an absurd amount of water, as well as the cell wall that is so rigid it's able to hold that water in and not burst. If it only had a cell membrane, it would burst. So I'm going to let you guys go through these activities on your own. Everything is basically written out for you, and there's the recordings are posted for you guys to actually watch two professors from RCC do the experiments. Activity 4, you would be observing both dialysis and osmosis. So dialysis is the diffusion of a solute. through a semi-permeable membrane. And to do this, you would use a dialysis tubing. And this dialysis tubing is what's going to represent your semi-permeable membrane. You're going to add this glucose starch solution to this dialysis tubing. So you'll pour it inside this little bag. And you basically have to think, what's going to happen? Do I think glucose is going to leave the dialysis bag, starch, or both? And you're also going to look at osmosis. So you're going to see, do you think water is also going to leave or not? And to do that, you're going to weigh your bag before and after the experiment. But the professors that conduct this experiment go through it, all these steps. But remember, this is a semi-permeable, representing a semi-permeable membrane, this dialysis tubing. So when you make this prediction, what do you think is going to be able to leave this tubing? 10% glucose or 10% starch? Remember that glucose is a monomer, it's a monosaccharide, whereas starch is a polymer, it's a polysaccharide. So this monomer is way smaller than this polymer. So just keep that in mind when you think about dialysis, diffusion, osmosis, things like that. Or just diffusion and dialysis, actually. So like I said, you're going to record the weight both before and after, and that's going to be to measure osmosis. You want to see, did water leave or did water stay in the bag? And then you essentially submerge it into a beaker, but they kind of go through it with you. There's some steps that aren't shown here, like you would need to actually rinse this bag after you fill it with the glucose starch solution. And you would want to stir this. So you stir it every 15 minutes. You leave it here for 2 hours for dialysis to occur. Once the experiment concludes, you rinse and dry your bag, you weigh it, and you see what happened to the weight. Did osmosis occur or did it not? And then you're essentially going to take 4 test tubes. I blocked out the numbers because I've noticed that the professors use different numbers than what your lab manual indicates, so I didn't want you guys to get confused. I think they just picked 2 test tubes when they were conducting the experiment for you guys at random, and your lab manual on page 48 indicates which test tube would have been used for which condition, but they're very clear about it. So you essentially grab 2 test tubes, and you're going to pour the contents of the inside of the bag into these 2 test tubes. And then you're going to pour the contents of the beaker, which is outside of the bag, into 2 test tubes. You're going to use different indicators to test for glucose and for starch. So in order to test for glucose, you're going to use Benedict solution. And Benedict solution is, it will turn a yellow-orange color if glucose is present. So you put the solution in, you place them in a water bath, a hot water bath for 15 minutes. You remove them, and then I think they microwave it in the video since they didn't want to wait the 15 minutes. And then you remove them, and we'll see that blue indicates negative, orange yellow indicates positive for glucose. And then this is actually a hazardous solution, and it can cause toxicity to aquatic environments. So you can't toss it down the drain, you have to put it in a waste bin. And then you will also look for starch, and to test for starch, you're going to use iodine. So iodine initially is an amber-like color. So you put a few drops of iodine into each, I think it's just like 1 drop of iodine into each test tube. And then to each test tube. And when you do this, it's to 1 test tube from inside the bag and 1 test tube outside the bag. And then if a dark blue color appears, that indicates that starch is present. If it retains this amber color, it's not present. So you'll want to make some hypothesis before you do your experiment, and then you also want to record your data before as well as after your experiment. And then activity 5 is going to look at osmosis in a potato. And once again, they're going to take you through this very carefully. They'll show you how you would collect your potato cores. But here you're going to expose your potato to different conditions. So like I said, you'd collect 4 equal pieces, soak them in water, and then you're going to put those potato cores into 4 different conditions. So you're going to have an isotonic solution, which is this 0% sodium chloride solution, which is just water. And then you have a 0.9% solution, 2.5% solution, and 5% solution. So here, the 0% solution is hypotonic relative to the potato. So you have to make a prediction. Since this is a hypotonic solution, what do I think is going to happen to the water molecules? Do you think water molecules are going to go into the potato or leave the potato? So you might be making your hypothesis that you think if you put the potato here, and since this is a hypotonic solution relative to the potato, that water is going to go inside the potato, so this potato will gain weight. So your 0.9% sodium chloride solution is considered an isotonic environment in relationship to the potato. So since this is isotonic, essentially you have to make a prediction. What do you think is going to happen to osmosis or the weight of potato? So this is an isotonic solution. So essentially water is going to be going both in and out of the cell, and it's going to be basically an equilibrium or homeostasis. So you can make the prediction that I believe they're going to be approximately the same weight since water may be moving in and out of the cell. There might be a slight increase or slight decrease, but it wouldn't be significant. And then the 2.5% and 5% solutions are considered hypertonic environments in relationship to the potato. So you have to make a prediction since they're hypertonic. What do you think is going to happen to the water molecules? You may say since it's a hypertonic solution, you believe water is going to leave the potato. So it's going to leave the potato, try to dilute that extracellular environment. And because of that, you're going to see a decrease in the weight of the potato. So you'll conduct this experiment. You're going to record the weight of the potato before and after the experiment. And like I said, the YouTube video goes through this. The professors conduct this experiment for you guys. So you guys will be there watching it. And that's essentially it for these activities. There is an extra activity in your lab manual that has you guys record cell structures and functions. I think it'd be very beneficial for you guys to do this activity, more so for your lecture portion, because it is important to know all the structures of different cell types. So if you guys have time, that'd be a great activity to do in preparation for your guys' exams. And don't forget, your guys' quiz is due on Sunday at 11:59 p.m. And that's it.